Ever found yourself lost in a sea of scientific jargon and intricate diagrams? Well, you're not alone. Reading biotechnology research papers can feel like deciphering a complex code. But whether you're a grad student or just someone curious about the latest advancements, we've got you covered. In this video, we're going to teach you the art of understanding scientific papers quickly so you can understand science directly from the source. If you've ever felt overwhelmed by the thought of delving into a research paper, you're in the right place. By the end of this video, you'll have the skills to confidently navigate biotechnology research papers. But in order to understand how to read a scientific paper, we first need to understand how a scientific paper is written. A scientific paper is broken up into several parts: abstract, introduction, materials and methods, results, discussion, figures, and references. In order to understand how to use each of these sections to their fullest, let's break down what information is in each of these sections. The abstract is a paragraph or two at the very beginning that summarizes the whole paper. The intro starts with a few paragraphs of background info, leading to a paragraph of what the researchers wrote about in the study. The materials and methods section often comes right after the intro, but sometimes it's located at the very end of the paper. The materials and methods section contains, or at least is supposed to contain, the detailed steps of exactly how the researchers did the experiments, including where they ordered their chemicals and supplies from. Ideally, other scientists could go back and just from the materials and methods section, repeat the experiments that the researchers who wrote the paper did. Sort of like a recipe. The results section sounds obvious, but what exactly are results? The results are a collection of data from experiments that the researchers feel fit together or are connected in some way. But just looking at the data on their own, it might not be obvious to someone else. The data is organized into figures, visual representations of the data like graphs, microscope images, and the like. The results section describes the figure panels in the order the authors feel fits together in a logical order. The discussion often starts off with a paragraph that summarizes the paper again, then generally a paragraph or so each on the following topics: limitations of the study, future research that could be done to build on the study, avenues of technology or research that the study adds to, speculation about additional things the data might suggest beyond what the researchers directly tested with their experiments, and so forth. So, in my first year at grad school, here's how I used to try to read a scientific paper. I'd start with the abstract. Reading just one paragraph to understand the whole paper is very tempting, and it seems logical at first glance. Then I'd read the intro to get all of the background needed to understand the paper. Pretty much skip the materials and methods entirely since it's so detailed and technical, then go through and try to understand the results line for line, and then go back and try to make sense of the figures, maybe scan the discussion, but by the time I got to the discussion, I was realistically too exhausted to spend much time on it. Can you relate? This way of reading a scientific paper just really was not very effective for me. But why is this? So the abstract isn't just a simple Spark Notes type version of the paper. Rather, it's a condensed version, which also means it often contains all the technical vocabulary, jargon, and complex concepts all condensed into one paragraph, with minimal context. This is the perfect place to get bogged down and start feeling like, maybe I just don't have what it takes to understand this. The intro does contain background, yes, but not necessarily the background needed to understand the paper. It's more a logical progression of ideas leading up to what the authors did. It usually starts with introducing a broad topic, societal problem, area of research to be developed, or other type of motivation for doing research in a general area. Then, the authors talk about how they came up with what they did. Other research studies are discussed that served as inspiration for the project, or were built off of, studies that left some unanswered questions that the researchers want to address. The background papers are of course included in the citations section, but before jumping down a rabbit hole of reading every research paper that's cited, let's look somewhere around the first sentence of the last paragraph of the intro. Generally, there would be sort of a thesis statement, starting out with something like, here we set out to, in this study we did XYZ, here our aim was, something like that. Understanding this key sentence is much more fruitful than trying to understand the entire abstract and introduction. So tip 1 is where to start. Find this key sentence. Sure, from one sentence we might not get all the info on where the researchers came up with their ideas and the societal implications and so forth, but we can at least get the main point of what the paper's about and define the key vocabulary. Next, let's check out materials and methods. If you're reading a scientific paper on a topic you're not really familiar with and looking to learn the science and research that was done, it really isn't the best strategy to read through the methods line for line. Imagine you've never done any cooking before of any kind and suddenly you're presented with a cookbook. There would be an overwhelming amount of numbers, measurements, temperatures, details, etc. Likewise, if you aren't used to doing similar types of experiments in the lab, the materials and methods section will likely be pretty confusing. But that doesn't mean there's no benefit you can get from this section. But instead of reading line for line and focusing on the stuff you don't recognize and all the numbers, let's point out some key items that can give you a clearer idea of what the paper is about. Like with our recipe example, even if you don't know all the details, you could still recognize some of the ingredients, like gluten-free versus regular flour, or cooked versus raw, painting a picture in your mind of the dish. The materials and methods is broken down into a series of subsections. Usually, among the first of these subsections will be some sort of description of the organism the researchers are using in their experiments, like cells and culture, clinical samples from people, or mice. Within this section, we can pull out some more info about what system they are studying, like 17 patients with kidney disease versus human kidney cells in a dish, versus mice with kidney disease as a model, or versus computer models simulating a disease trajectory, etc. So tip 2 is scan through the materials and methods and look for what types of animals, samples, or systems the researchers used. Next, let's go to the results and figures. These sections go together. The figures are basically the data from the experiments, while the results are the author's description of the figure panels and where they point out things that are especially important within the figures. When I first started reading scientific papers, I used to read the results line for line and try to understand it and then glance at the figures almost as an afterthought. The results and figures make up the main content of the paper, and it's also an easy place to start feeling overwhelmed. There's a saying about writing scientific research. Your data should tell a story. Now, it isn't really a story in the conventional sense, like telling the day-to-day happenings in the lab, but rather it means organizing the data in such a way to form a logical progression of ideas that form support for some hypothesis, conclusion, statement, or so forth. And like a story, a scientific paper is generally broken up into sort of chapters, one for each figure. Often, these chapters have either a bold or italic heading, making them easy to find. The figures are further broken up into figure panels. Each panel usually contains the results of an experiment. Think of these panels like paragraphs within the chapter. And generally, for each figure panel, there's an accompanying descriptive sentence or paragraph stating the author's description of that panel. But there's another piece of information that's often overlooked, the figure legend. Why bother with a figure legend if all the panels are described in the results? Although both the figure legend and the results describe what's in the figures, there's a key difference. The results describe the author's interpretation of what they see in the figure panels and their conclusions based on the data. The figure legends state in a more unbiased way what is being shown without any conclusions or interpretation necessarily. This can actually make the figure legends an easier place to start than reading the results section line for line. The figure legend usually has a title for each figure followed by a brief description of what is being shown in each figure panel. So tip 3 is to go back and rewrite the titles of the figures in your own words. This might be easier than it looks at first glance because you've defined all of the acronyms and jargon needed to understand the key sentence we identified in tip 1, and these terms are often used again in the figure legends. So now that having been said, the figures themselves can be a bit overwhelming. Often scientific papers have anywhere from 4 to 6 or even more figures, each with anywhere from a few to up to a dozen or so individual figure panels within them. It makes sense then to focus on the really key figures, the experiments and data sets that played the biggest role in the paper first. Now, it isn't a hard and fast rule, but I can tell you from experience that usually the most important figures in a paper come more towards the beginning, not surprisingly. The first 1 to 2 figures. Then generally, many of the figures after that will more be used to support or provide additional evidence for the hypothesis that the researchers proposed to explain the data they found in the first figures. Now, this doesn't mean that the majority of the figures are just unimportant. But to get the main point of a paper and understand it most efficiently, it makes sense to start at least with the most key data. So, tip number 4 is using the titles of the figures that you've written. Pick out what you feel are the most critical figures to understanding the paper, keeping in mind that often these will be amongst the first figures to appear in the paper. But now that we've identified 2 key figures that we really want to understand, let's break them down further into key data panels. But instead of going through the results that correspond to the figures line for line, let's take a different approach. Look at the figure, say we're going to start with figure 1, and take each figure panel and identify something of interest to you or a trend you see with your own eyes. Perhaps an increase in one group presented in one of the graphs, or a schematic showing the author's experimental setup, or a bunch of data points, or a trend line. Now write out a short sentence describing each figure panel in your own words. Even if you don't understand all of the figure or the methods used to generate it, that's okay. You don't need to be an expert to see data with your own eyes and start forming opinions. That's tip 5. Now let's go back and see what the authors say about their data. Each figure panel in the results section should have a sentence or so that corresponds to it. So try to take each of these sentences one by one and rewrite them in your own words. What are the authors trying to say about what they found? There may be jargon and terminology that you don't know, but try to identify vocabulary words that appear multiple times throughout the figure panels that you're looking at. Just like when you're learning a new language, you don't necessarily need to understand every single word that's said in order to understand the meaning overall. Scientific literature is just a new language. So that's our next tip. Do your best to write out in a short plain sentence what the author's observations are on each figure panel out of the 1 or 2 figures you've picked out that you feel are really key to your understanding the paper. Ask yourself, are there differences between what you observed and what the authors observed? After hearing what the authors have to say about their data, do you agree with it? Can you follow what they're saying? Do you feel that the authors focused on the most important portions of the data? After reading what the researchers have to say, did any of your opinions on what you see in the data change? And there's not necessarily a right or wrong answer to any of these questions. The purpose of scientific writing is communication, to open a conversation about data that's been collected. It gives researchers a place to explain how they feel their data fits in with current research and add something to our body of existing knowledge. But these are still opinions, even if educated ones. Scientific interpretations are continually debated, refined, and improved upon. The discussion section of a paper is a place where the researchers can share their views and speculate on how their data fits into current research and technology in the future, and discuss areas they feel would be of value for future research. The discussion is different from the results section in that the results are limited more strictly to the author's views that can be directly supported by their experimental data. The discussion, on the other hand, can be more subjective, But that doesn't mean the discussion is totally not worthwhile. But instead of just reading the discussion line for line, our next tip is to look for the following points in the discussion. First, what additional research do the authors see as being important for the future in this area? And two, what do they admit are current limitations of their study, or gaps that they were not able to fully address here? Reading with this purpose in mind gives us a much easier way to stay focused when reading the discussion, instead of getting caught up looking through tons of information. Often, in the very last paragraph of the paper, the authors share their view on what they feel is the impact or the value of the research in the field. Somewhere in the middle, there should be a paragraph regarding the author's views on the limitations or gaps in their study. Try to write a sentence for each of these in your own words. Finally, let's discuss an overlooked section of a scientific research paper, the citations or references section. The citations or references are basically the other scientific studies that the researchers built upon when performing the current study. If you find that you're interested in learning more about the type of research done in a paper, it can be valuable to pick out some additional papers from the references section to go back and read. So our final tip is don't forget to check out the references section to see if there are any additional papers in a particular area of study that you'd also like to read. I hope you found this brief video guide to reading scientific papers in the field of biotechnology useful. The mission of this channel is to make real scientific research available, accessible, and fun for everyone. To support our mission, please give us a like, subscribe below, and share with your friends. Thanks for watching, and remember,